Good evening. My name is Karen Ilinich, and I have the privilege of welcoming you to the Franklin Institute. This evening's program continues our tradition of providing public audiences with access to extraordinary science education. We are delighted to again welcome the Penn Genome Frontiers Institute as our partner in health science education. It's a great time to be at the Franklin Institute. We are currently hosting a world premiere exhibition called Cleopatra and the Search for the Last Queen of Egypt. If you haven't visited yet, I highly recommend that you do so. It's a beautiful and inspirational exhibit made possible by a careful and methodical underwater excavation in the port of Alexandria on the northern coast of Egypt. The magnificent artifacts that are on display upstairs have been buried beneath the sands for hundreds of years, only recently brought to the surface. The exhibition will be here through January 2nd. It's also a great time to be in Philadelphia, as science organizations throughout the city are coming together to plan the first Philadelphia Science Festival, which will take place from April 15th to 28th of 2011. There will be a day-long carnival here outside on the Parkway and a program of events happening at venues throughout the city. The goal is for every Philadelphian in every neighborhood to get excited about science during the festival. The festival will culminate with the Franklin Institute's Awards Week programming, and the medal presentation will happen on April 28th. The focus this year, coincidentally, is on genomic science, as the Bauer Award will be presented to a leader in the field. Stay tuned for more information as the details for both the festival and Awards Week become finalized. Be sure to mark your calendars for April 2011, which promises to be a great time for science in Philadelphia. Now, it is my pleasure to introduce our co-host, Dr. Eberwine, the co-director of the Penn Genome Frontiers Institute.
Thank you, Karen. I'm Jim Eberwine. I'm one of the co-directors of the Penn Genome Frontiers Institute. I work with my colleague and friend Junhyong Kim, who is going to be one of the speakers tonight. And I wanted to welcome you all to this symposium. This is the fourth one that we've done with the Franklin Institute, and they've been great partners, and it's been always an exciting evening and filled with interesting science and interesting questions from the audience. So the way in which we like to do the symposia is that we'll have a moderator who I'll introduce in just a few minutes, who will introduce the speakers and keep everyone on time. Then we'll have a panel at the very end where the speakers will be joined by two other experts in this area to answer questions that the audience poses. So throughout the talks, we would like for you to ask questions, write questions down on cards, and you'll see it up here on the first slide, and give them to one of the PGFI representatives. And then our moderator will select some of those for the panelists to answer. So what I thought I would do is give you a little bit of background to prepare you for the talks today. So one of the questions that I'm often asked in the classes that I teach is, what is genomics? And so genomics is a term that a lot of people might define differently, but for me, genomics is a study of the genomes and RNAs of organisms. And the important component of this is a transformative idea because it's not a single gene, but rather multiple or even all the genes that are analyzed in genomics. And it's a transformative idea in biology because up until the idea of genomics, people would study one gene at a time or one protein at a time. And genomics tries to enable people to look at multiple ones simultaneously because that's how cells work, and that's systems biology. So this is a DNA, and this is what you'll be hearing today about from several of our speakers. This is a double helix discovered by Watson and Crick, and this is one base paired to another base. And you'll hear more about this momentarily. But I use this as an introduction to highlight the fact genome comes from the Greek meaning I become, I am born. That's what genome means. The first genomicist was Fred Sanger, and he became the first genomicist when he first sequenced 5X174. And when I say sequence, it means looking at the DNA structure. You're going to be hearing more about this momentarily. The sequence of this genome, the first one done in 1977, was about 5,400 bases. Now we know that, you're going to again hear more about genomic DNA structure, but all humans share DNA. The similarity in the DNA is quite high between individuals, but it's these differences in DNA sequence that give rise to individuals. With that said, looking at the field of genomics, the Penn Genome Frontiers Institute is an institute at the University of Pennsylvania. It's a Provostio Institute, meaning that we're under the Provost's office. There are over 100 members from around the university who are members of PGFI. And the mission for PGFI that Junhyong and I have set is to try to be an interdisciplinary institute bringing the various schools together, engineering, the vet school, arts and sciences, the med school, and even Wharton, to try to solve various problems in genomics, bringing technology and ideas to the field to try to promote genomics around the campus and around the country. One of our initiatives that you'll be hearing again a lot more about today is sequencing. So when you think about genomic science and genomic DNA sequences, you need to be able to identify each of the bases in the sequence. Again, you're going to hear more about this in detail momentarily. But there are new types of instruments called next-generation sequencers that allow us to do this very rapidly. When Fred Sanger did the 5X174 genome, it took him nearly 3 years to do 5,400 bases using a technique that he developed for sequencing. Now, with one of the next-generation sequencers we have at PGFI, we now have 3 of them, we can do 45 billion nucleotides in 1 week. So we have ramped up the speed at which sequencing can be done very rapidly, and we have a lot more detail that can be generated very quickly. Where this is important for tonight is, as we think about microbes, there are a number of bad microbes. So one annoying microbe is a fungal microbe involved in an athlete's foot. A bad microbe is the HIV virus, which you can see here, infecting the red blood cell. Of course, a cholera virus, or cholera bacteria, which we're all hearing about with respect to Haiti and the terrible problems that are ensuing as a result of the problems in Haiti from a few months ago. And of course, there are very good bacteria, like this bacteria that's in your gut that's involved in keeping other bacteria from growing by virtue of what it's involved with. We're all tied together with it with respect to this through our DNA. Each of these organisms has DNA. We have DNA, and it's the interplay between these genomes that gives rise to the bacterial flora and microbial flora that survive within us and can help us and can be detrimental to us. So with that as a background, our moderator tonight is going to be Ivan Oransky. He's an executive editor at Reuters Health. He's also an assistant clinical professor at NYU, where he teaches in the journalism school. Dr. Oransky has worked with various scientific publications over the last several years, including being the online editor for Scientific American before he went to Reuters. And he will be our moderator who will lead the rest of the discussion.
Thanks very much, Dr. Eberwine, and thanks to everyone for being here. I know I'm looking forward very much to the discussion, to the presentations, to learning a great deal more about genomics, a subject that I am somewhat inadequate in, as many physicians are, and I hope I don't offend any physicians in the audience. I want to say just briefly a little bit about how we will be structuring tonight. Obviously, there will be 3 presentations, and then, as Dr. Eberwine mentioned, there will be a discussion after that with a couple of other experts. During any of those times, please feel free after each presentation, and then obviously during the discussion as well, to write down any questions you have. There'll be note cards circulating that you can do that on, and then at that point, I will be selecting some questions, and really any are fair game, and we appreciate all of them. So first, I want to introduce someone who Dr. Eberwine has already mentioned, Dr. Junhyong Kim, who is the Edmund and Louise Kahn Endowed Professor of Biology and co-director of PGFI, so many of you are probably familiar with his work. He carries out research in genomics and computational biology, and his research programs are also in systems biology, comparative neuroscience, and theoretical biology. He served on a number of advisory groups and editorial positions, including the NAS, Koshland Museum Advisory Board, and others. And he has won, as you would imagine, a number of awards, including a Guggenheim Fellowship, Ellison Foundation Senior Scholar Award, and the Sloan Foundation Young Investigator Award. Dr. Kim.
Thank you very much, and thank you all today for coming out. So, genes are our destiny. However, actually, of course, genes are not our destiny. Our genomes are not our destiny. As, in fact, as Jim alluded to earlier, if you take all of human diversity and some of the wonderful diversity that Dr. Tishkoff is going to talk about today, 99.7% of our genomes are identical. So we are pretty much the same when we look at our genomes. However, despite all this, of course, we all have our specific traits, our specific functions, our specific looks. So in fact, our genomes do what little bit that's different from each other do determine to a large extent or influence how we are. So here, for example, is my son who shares 50% of my genome and also shares with me this terrible trait of getting migraine headaches. Of course, he shares the good looks with his mother, for which I'm very grateful. And this arises, of course, out of the genome that's inherent in the small single cell from the first egg and the sperm coming together, propagated through all the cells in our body. In fact, we have 100 trillion cells in our body. And if we looked at any one of these cells and looked inside, what we will see is a set of chromosomes. And each of these chromosomes contain strings of DNA. And if we look very closely, what we will find is that each of these chromosomes contains strings of DNA, which are tightly wrapped around each other and packaged into this tight package that we call chromosome. And each of these strings contain actually letters. And what we say letters is actually a metaphor for individual units of molecule. And so we have 4 letters, A, C, G, T, which are individual molecules that are tied together in a long string. And one of the important parts is that the particular sequence of these letters is important. And just like the English letters, the sequence matters. So if we have BAT, B-A-T, this is certainly different from TAB, T-A-B. And similarly, the letters A-C-T in our genome is different from, meaning to the organism, different from T-C-A. And therefore, this is the reason why we want to learn the sequence of the genome. And the sequence of the genome means something to the organism, and it also means something to us. So just to reiterate, the genome consists of these 3 billion DNA letters in our entire genome. Physically, if we were to take all of these genomes and stretch them out together, in each cell, there's about 6 feet of DNA information packed into each cell. And since we have hundreds of trillions of cells, if we were to link all of these end to end, what we will find is that we can span the distance between Earth and Moon 50 times. So this is the amount of information that we all carry around in our body, in our genome. So the genome, the 3 billion letters of information, then specifies each cell and tells each cell what to do and to make tissues in our body. It tells the tissues to make organs. It tells the organs to make us. And that is what genome specifies for us. So the genome is sort of like an instruction book for our bodies. The problem is that this book is rather cryptic, and so it's written in a language that we don't really understand. We know only a few words, maybe a little bit of punctuation. We don't really know where the stops and the starts are. And we know some sentences here and there, and this is what we have to work with. So genomics is really all about learning to read this book and figuring out what the book tells us about our bodies. So over the last 40 years, biologists have figured out how to make more DNA, copy DNA, break DNA, manipulate DNA. And we learned all of this because what we did was co-opt lots of existing tools from nature. We took tools from viruses, bacteria, jellyfish, worms, and so on. So one of the hopefully key messages that you take home today is, in fact, even when we're interested in things like diseases and biomedical sciences, without such advances in fundamental biology, we would not be able to have the whole suite of toolkits that we have today in order to work with and to try to design therapeutics for our own diseases and our own bodies. So an important aspect of this genome, which I hope to tell you a little bit today, is in fact these molecules, nucleotides that we just talked about. So nucleotide comes in 4 flavors, as I said, A, T, C, G. And one of the important aspects of this is that they have certain attractive forces to each other. So A likes to stick to T, and C likes to stick to G. What I mean by that is they sort of act like magnets inside our cells. And so by biophysical forces, if you put them close, they'll tend to stick together. And this, we call this base pairing. And because of this, we also sometimes call these letters base pairs. And this base pairing really is the fundamental basis of how information is copied in our genome and transmitted through a different cell. So if we have, say, a string of DNA, which I've symbolically depicted as, say, a ball, a cup, a square, and a square cup, and let's say this is ACGT, what will happen is that if we have another string that is what we call complement to this string, and you'll see what that means in a moment, that where there is a ball on one side, there is a cup on the other side, where there's a cup on one side, there's a ball on the other side, they will come together and tend to stick together. So what we have is information transfer from the string on the left side, string on the right side, by the fact that they are able to stick together. So this is, in this case, we say that we have formed a duplex, and this is really the basis of many of the biotechnologies that we use, as well as how information is manipulated in our cells. So just to give you an example, how do we actually sequence the genes in our genome? Well, we'll start with some potentially unknown string. So to make it unknown, I put a black box in front of it. And imagine that we have now a set of boxes full of nucleotides. Those were the letters that I talked about. And I have 4 boxes. And using some chemical magic, I put a little light on each one of them. So I put orange light here, green light here, red light here, and blue light there. What will happen is that we go and we try out each of these lights and stick them to the unknown string and see if it sticks. And so we try the green one, we try the red one, until we find one that sticks. And when it sticks, what we get is a little light turns on. And what we want to do is have a little movie camera and take the picture, a movie of these lights turning on. So we'll get the red light, green light, and the orange, red, red, and we'll have taken the movie, and eventually we play back that movie and we see blue, red, green, orange, red, red. And that allows us to realize, in fact, what was under this black box. And all of this happened because we had these base pairing interaction that only allowed this blue one to go right there, red one to go out there, and so on. So as Jim mentioned, well, in 1980, I first started doing science. And at that time, it took us about a whole year to obtain about 600 letters of information. That's about 1.5 letters a day. Jim already showed you these next generation sequence machines. This is the whole idea of genomics, which happened about 15 years ago, where we started designing new machines and new measurement technologies which were faster, smaller, and more precise. And this has just become an exponential growth over the years. So when I first started doing work, we started working with experimental systems like this, which is, everybody's familiar with test tubes. These are kind of small test tubes, not that small. About 10 years ago, we were able to make devices like this, which in the area about size of a quarter, you can carry out 1 million separate experiments. So these were manufactured using the kind of techniques used to make computer chips. And now, in 2010, we have devices where we can do 1 billion experiments in the same quarter area. And so by making things smaller, we're able to generate a lot more data and a lot more fastly. So as Jim said, now with the next generation sequencing, we can generate 20 billion letters per day. So just to recap, in 1980, we were able to do 1.5 letters per day. In 2010, we are able to do 20 billion letters a day. So this is about 13 billion-fold increase in efficiency. And if you're an investor, this is a compound interest rate of 230% every year for 30 years straight. So this is how you become a billionaire, starting with $1. And so if you want to invest in something, please invest in science, because nobody gives you return like science will give you. So right now, we have the capacity worldwide to generate 4 trillion letters of information every day. And pretty soon, all of this genomics will become diagnostic. So when you go to the hospital, within the next 5 years, we will be sequencing your genome just as you might take an MRI. There will be 1 billion genomes that will be sequenced around the world. If we had 1 million scientists whose job is to do nothing but stare at the DNA genome sequence that's coming out, we will get maybe about 0.5% of the information looked at by human eye. So vast majority of the information out there cannot be seen or handled by humans. So the only thing we can do, of course, is to use machines, and the machines are computers. So I am a computational biologist. What we do is use computers and mathematical tools to try to understand all of this genomic information that is coming out. So the one take-home message now is that in this modern era, right now, we cannot do anything in biology or medicine without utilizing computers and mathematical models. So in the last 20 years or so, computational biologists have learned how to do all kinds of things with DNA sequences, arrange them in an informative way, find specific motifs that confer function in the cell, and so on, by using now tools from lots of different fields, including mathematics, computer science, statistics, physics, cryptology, internet, even computer games, which my son is playing right now. So, I can talk about a lot of different things, but today I wanted to talk about just one little aspect which will be relevant to some of the next talks that you hear today, and that is how do we use the genome to predict genealogies of organisms. So all organisms arose through the specific process of making copies, so making 2 copies and splitting, and having each copy become modified. So Darwin called this descent with modification. So all biological objects, whether they're genes or whole organisms, arise through this kind of descent, bifurcating descent with modification process. And so the particular structure, this tree that we've drawn here, is a genealogical tree of this history. And so we call this tree of phylogeny. That's just a technical term for this genealogical tree. So what I want to talk about today is I'll give you a little bit of flavor of what we do in order to get such trees from data. Well, first, what do we do with such trees? Well, for one, you can learn very surprising things about organisms. This is a lotus flower. This is a water lily, and these two are beautiful flowers, quite different from this oak tree here. But if you knew actually the genealogical history or the phylogeny of these organisms, what you would learn is that the lotus flower is actually more closely related to an oak tree than it is to a water lily. Of course, genealogy or evolutionary history doesn't mean just the ancient past. Things like viruses are fast evolving and leave traces in everyday transmission. So here's a case in which West Nile virus outbreak was first noted in New York City in about 1999. And by looking at their genome, we were able to trace the West Nile virus to another strain that was an Israeli strain which had an outbreak in 1998 that allows us to design or find out some more clues about the origin of disease as well as the prognosis of the spread. There are also cases in which influenza viruses are changing every year, and that's why we have to get new vaccination every year. And we can try to predict their evolution using these phylogeny techniques and design vaccines in the previous year before the new strain comes aboard. So there are many wonderful things we can do with phylogeny, but of course the most important part is it reveals to us the wonderful biological diversity we're normally not aware of. So for example, there's you, and then there's your dinner right here. And most of the time, we are only consciously aware of this part of the biological diversity. But of course, the biological diversity, this is only a tiny fraction of biological diversity. And we have all of this diversity, some of which Dr. Bushman will talk to you today. So how do we infer these genealogies? Well, suppose we have 4 organisms whose genealogy we'd like to trace. There are many, many ways to do this, but there are some simple ideas we can apply to this. One idea is to simply split these organisms by some shared trait. So for example, some organisms have backbones, other organisms don't have backbones. Of course, depending on your inclination, you might think this should be like this or like this. And this is one reason you have to be very careful when you assess somebody's character. Now, of course, once you have such a split, you might do this a little bit more and say, okay, lays eggs, don't lay eggs. Has hair, don't have hair. And when you make this split, it naturally suggests to you that this should be the phylogeny or the genealogical history of these organisms. And the main idea here is that any shared trait that they have, say, have backbone, is due to evolution of the trait along this particular lineage, and they have the trait because they all share that particular history. Of course, we can turn this into DNA and do exactly the same thing. Instead, this time we might say, okay, look at gene 1 and ask, is there a letter A or is there a letter C? Look at gene 2 and ask, is there a letter T or letter C? Gene 3, look at, is there a letter A or letter G, and so on. So the main advantage here is instead of looking at a small number of traits, we have the potential to look at millions of such letters because we can get the whole genome sequence, and that allows us to make more precise and more detailed inferences about these genealogical trees. So we can get these with fairly simple arguments, but the problem is something like this. What happens when you have one of these things, a platypus? Okay, so here's an organism with hair, lays eggs, and has a bill. So where do we place this organism? You can think of many different ways to do this. One idea that's been generated, well, let's consider various different possible phylogenies and see if it implies any kind of contradiction. So if we put the platypus over here, well, lays eggs and has bills. Well, this is a bill too, I guess. Well, thank you for that. Well, we have a contradiction with the hair not sharing together. Right? If I put it over here, now we have 2 organisms with hair, but no eggs and no bill. So we created 2 contradictions. So it may be best that one want to minimize and choose the phylogeny that minimizes such contradictions. Okay? Now I've simplified this. In practice, one can have thousands of such contradictions, so it becomes a little bit hard to compute. But regardless, it's a fairly simple idea. Well, so what's the real problem? Well, if you look at 3 items, then there's about 3 different genealogies. So here's 3 possible ways in which these organisms can be related to each other. I like the one that puts John Stewart and the platypus together. But if you have, say, 4 items, turns out there's 15 such possible phylogenies. 5, there's 105. If you have 10 items, there's 34 million possible genealogies. If you have 30 items, there's no more possible genealogies or phylogenies than there are atoms in the universe. So what do we do? Well, that's why there's people like me, computational biologists. And so we develop efficient algorithms and new computational tricks so we can do small phylogenies, medium-sized phylogeny. This is a tree that's been wrapped around just to give you a more better visual display, or very large phylogenies with very many items. In fact, this is another phylogeny where each individual organism looks like stars in the universe. This is visualized in what's called a hypergeometric sphere. And each one of these little dots here, which you don't see, represents an organism. So as computational biologists, we got this handed. And that's why we get the big bucks. So in conclusion, genomes strongly influence organismic traits, all of our forms and functions, disease risks, and so on. We now have the ability to really efficiently read the genome information. And the key is to learn the meaning of all this information, for which we still only know a tiny fraction of the words and how it goes. And this cannot be done without computation and modeling. And this is why mathematics, computation, statistics has become integral to biology today. So let me just finish with one last thought, which is that some people say biology is the youngest of the sciences. And all natural sciences, like physics and chemistry and astronomy, all went through these stages where we started enumerating what's out there, what are each individual object, into starting to theorize about what is the organismal or organizational structure of nature. So now we have the ideas about how orbits of planets work, what the structure of the galaxies are. And biology really has just gone through this transformation with the huge amount of volume of data that's being enabled by genomics. That's why we're so excited. We feel that genomics is finally beginning to allow us to get real handle on the complexities of the organisms that we are today. Thank you very much.
Thank you, Dr. Kim. So if I could sum up, if we invest in science more, we will be able, because of this tremendous compounding, to actually understand whether a platypus is more related to Jon Stewart or Bill O'Reilly. Sorry, just doing my journalistic, bringing it down to some level there. Thank you very much. It's my pleasure to next introduce someone who's going to bring things into the human realm a little bit, and maybe even tell us exactly who's related to who there, leaving out the platypus. Dr. Sarah Tishkoff is the David and Lynn Silfen University Associate Professor in Genetics and Biology at the University of Pennsylvania. She also holds appointments in the School of Medicine as well as the School of Arts and Sciences. She's a leading global expert in human genetics. She works primarily in Africa, where she's compiled an extensive DNA database. Her research examines how genetic variation can affect a wide range of practical issues. For example, why humans have different susceptibility to disease, how they metabolize drugs, and how they adapt through evolution. She's won a number of awards, including a 2009 NIH Pioneer Award, and she was also named one of Popular Science magazine's Brilliant 10 American Scientists in 2003. So please welcome Dr. Tishkoff.
So thank you for that introduction, and I'm really pleased to be here today to tell you about my lab's African Odyssey through the human genome. So it's thought that the human lineage diverged from the chimpanzee lineage somewhere around 5 to 7 million years ago. And in fact, we're remarkably similar at the genetic level. If we compare the human genome sequence to the chimpanzee sequence, we differ at only about 1.5% of the sites. And so some of the questions that my group, as well as other groups, are interested in is to try to identify the genetic variants that differentiate us from our closest relative, genetic relative, the chimpanzee, and from each other. And in particular, we're interested in the genetic variants that differentiate common traits like height, and those that may play a role in susceptibility to disease. So I want to tell you a bit about modern human origins. It's thought that anatomically modern humans originated in Africa approximately 200,000 years ago. We see the earliest fossil evidence for anatomically modern humans in a site in Ethiopia called Omo. But it was not until around 50,000 to 100,000 years ago that one or a few small bands of humans migrated out of Africa and populated the rest of the world. And it's thought that there may have been two primary migration routes. There was one that was across Southeast Asia into Australo-Melanesia, where we see the oldest archaeological evidence for modern humans dated to around 40 to 60,000 years ago. There's no evidence for modern humans in Europe until about 40,000 years ago. And in fact, we overlap for at least a good 10,000 years or so with Neanderthals and possibly other archaic species that were outside of Africa. Now, all of those other species disappeared for reasons that we're not entirely sure. And then there were more recent migration events. such as the migration into the Americas across what was formerly the Bering Strait, sometime between about 15,000 to 30,000 years ago, and more recent migration events, for example, into the Pacific Islands. Because we all share a very recent common African origin, as Dr. Kim was saying, we're remarkably similar at the genetic level. If I were to compare any genomes between any two of us in this room, we would differ at only about 1 every 500 to 1,000 letters. And so that's really remarkably similar. Now, one thing we're still trying to determine is the amount of structural variation in the genome. So that means regions where there's been insertions or deletions or rearrangements, and we haven't yet characterized that. But nearly every study that's been done that compares genetic variation in different populations has shown that the majority of genetic variation is within populations, and only about 10 to 15% is between populations. Now, the type of variation that I'm going to tell you about today consists of either sites in the genome where individuals differ at a single letter. So, for example, one chromosome may have a T, the other may have an A, and we call that a single nucleotide polymorphism, or SNP for short. The other type of variant is referred to as a short tandem repeat, or a microsatellite polymorphism, or variable site. And these are simply short segments of DNA letters, such as CTA, that are repeated multiple times and differ in terms of the number of repeats among individuals. These tend to have a very high mutation rate, and they're often used for forensic purposes. The other type of variant I'm going to tell you about are insertions or deletions of anywhere from 1 to could be up to 1 million letters in the genome. And these tend to be very stable when they occur. So this slide shows you some of the individuals that my lab has studied in the past 10 years. And you can see that there's quite a bit of variation, cultural variation, linguistic, and I'll show you genetic variation as well. And we're interested in trying to identify the genetic and environmental factors that play a role in normal variation and variation in susceptibility to disease. So I need to give you a little bit of background information about Africa. There are over 2,000 ethnic groups in Africa, and they speak languages that have been classified into 4 major language families. So in blue, we have Afroasiatic languages that are found mainly in North Africa and East Africa, and these would include, for example, the Semitic languages, spoken also in the Middle East, and Cushitic languages in Ethiopia. In red, we have the Nilo-Saharan languages, and those are spoken mainly by groups like the Maasai, pastoralist groups. The most widespread language family is shown in orange. That's the Niger-Kordofanian language family, including one of the most common language groups, the Bantu languages, which are thought to have originated in Nigeria or Cameroon about 5,000 years ago and then rapidly spread across Africa. And the last language family, shown in green, consists of languages that have clicks. And that's mainly represented by the San Bushmen in southern Africa. If you saw The Gods Must Be Crazy, you might remember that. But there are 2 other groups that speak with a click, called the Hadza and the Sandawe, who live in Tanzania. And their relationship to the Southern African San still remains to be determined. So this is why I think it's so important to embark on this African Odyssey. For one, if we want to learn more about modern human origins, we need to be looking in Africa. Secondly, if we want to learn more about African American ancestry, we need to be studying ancestral African populations. Third, we know that there's a very high incidence of infectious disease in Africa, with malaria, TB, and HIV being the 3 biggest killers. If we want to understand the environmental and genetic factors that play a role in susceptibility, we need to be looking in Africa. We also know that there are a number of complex diseases, meaning those that are caused by environment and multiple genes, that are very common in populations of recent African descent, such as prostate cancer, hypertension, and diabetes in African Americans. And again, a knowledge of variation in ancestral African populations will be informative. And lastly, we know that people differ in regards to response to drugs. And that's probably due largely to variation in drug metabolizing enzyme genes. And we know very little about this in Africa. And in fact, Africa has been greatly underrepresented compared to other regions. Now, why is that? Part of it is simply a lack of infrastructure and the challenge of doing research in that region. And so what I wanted to do was show you some slides giving you an idea of what it's been like to actually do the field work that we do in Africa. So for the past 10 years, I and my grad students and postdocs and African collaborators have been studying mainly minority populations in Africa. And these are groups that practice an indigenous lifestyle, and they live in fairly remote regions. Most recently, 2 postdocs in my lab, Simon Thompson and Alessia Ranciaro, just completed 5 months of field work in Ethiopia. And I like this picture because you can see the outside of the Land Rover. We had to bring all of our supplies, and then you get the inside perspective as well. And then you can see some of the challenges that we faced. A lack of paved roads, rainy season, a lot of difficulties. This is one of the groups of the click speakers that I told you about, the Hadza, who live in Tanzania. So we are very careful to get permission at multiple levels, at the government level, the regional level, the village level, and ultimately informed consent at the individual level. We also get detailed ethnographic information, and we get detailed information about a number of variable traits, including, for example, height and weight. This is from our recent trip to Ethiopia. We looked at blood pressure. We looked at a number of other traits that play a role in cardiovascular traits or metabolic traits, as well as skin pigmentation. We looked at lactose tolerance, taste perception. We collected blood from these individuals. That was an individual from the Maasai group. This is a group called the Surma from Ethiopia, who speak a Nilo-Saharan language. And we had to, we're actually collecting saliva in this case, and you can see what the conditions are like when we try to process the samples. And in fact, in many areas, we don't even have electricity. So here's a case where we had to hook up the centrifuge to the car battery. And typically what we would do to get DNA is to break up the red cells and spin down the white cells that contain the DNA. And then we have to process them. And again, very often we don't have any electricity, but we can either bring a generator or use a generator, and we essentially set up our labs on site. So what I want to tell you today is about a study that we published last year, which characterizes genome-wide variation in a diverse set of Africans. And we looked at approximately 800 of those short tandem repeats, or microsatellites I told you about, and 400 of these insertion-deletion variable sites in over 3,000 Africans, 98 African Americans, and 1,500 comparative non-African individuals. And here you can see a phylogenetic tree that you just heard so much about from Dr. Kim, in which we calculated genetic distances between populations. So if 2 populations are genetically more similar, they're going to cluster together in that tree. You can't see any details here. But what I want to point out is that the populations tend to cluster by major geographic region. So I can't see it, but if I recall correctly, we have India up here. I can hardly see. I believe Europe is here somewhere. We have Americas, Oceania, and then we have Africa here. And even within Africa, they cluster by major geographic region. So we have North Africa, East Africa, Central and West Africa, and Southern Africa, with one exception. The Pygmies, who are from Central Africa, are clustering with the South African San. And I think that indicates shared common ancestry, which I'll get back to. The other thing I've done is classify these based on the languages spoken and then color-coded them. And you can see that populations typically cluster with other populations that share a common language, but with a couple of exceptions. One of which are these languages shown in blue here. And these languages are called Chadic languages, spoken in northern Cameroon and Chad. They're classified as Afroasiatic, but they don't cluster with any of the other Afroasiatic populations. They actually cluster more with Nilo-Saharan. And we think that was due to a language switch that occurred within the past 10,000 years. The next thing I want to show you is inference of individual ancestry. And what we do is to basically, based on those genome-wide markers, for each, we actually infer the number of ancestral populations represented by different colors. And this is actually composed of many lines, where each line represents an individual, and the color represents their proportion of ancestry in that ancestral population. So for example, here are the non-African individuals, and we can see that people who self-identify as European or Middle Eastern tend to cluster together. These are individuals who self-identify as Southern Indian, East Asian, Oceanic, Native American. These are individuals who self-identify as Pakistani and Central Asian. And the point I want to make here is just look at all the colors in Africa, a tremendous amount of diversity, and it's clear that there's no such thing as a single representative African population. Just to point out a couple of trends, in orange are the populations that speak a Niger-Kordofanian language. In purple are those that speak Afro-Asiatic languages and are thought to have come from Ethiopia. In red are those that speak Nilo-Saharan languages coming from southern Sudan. And then we have groups like the hunter-gatherers, the Hadza, the Sandawe, the Pygmies, and the San. If we repeat this analysis just in Africa, and then to simplify things, we've pooled individuals together and shown them as these pie charts. Again, you can see a lot of variation, even within small geographic regions like northern Tanzania. And we can also trace historic migration events. So for example, in orange are the people who speak a Niger-Kordofanian language, and we see evidence of the Bantu expansion from West Africa to East Africa, where they admixed with indigenous local groups, and into Southern Africa. And we also see some other interesting things. Here are the Central African Pygmies, and then in lighter green are the South African San populations. And we see some evidence for common ancestry of the most eastern Pygmies with the San Bushmen. And in fact, we see several lines of evidence that suggest that several of the major hunter-gatherer groups in Africa share an ancient common ancestry. Now, interestingly, 3 of these groups speak with click languages. The 4th one, the Pygmies from Central Africa, lost their indigenous language. We have no idea what it was. But we hypothesize that maybe they too spoke with the click at one point. So it still remains to be determined exactly when these populations split, but it's very intriguing to think they may have had a common ancestry. And then 2 of the groups that we saw that had the most mixed ancestry included African Americans and self-identified Cape-colored population from Cape Town in South Africa. The African Americans that we studied, as you can see by this orange ancestry, they had predominantly West African Niger-Kordofanian ancestry, consistent with the history of the slave trade. But the most variability that we see is the amount of European ancestry, which ranges from 0 to greater than 50%. We also found small amounts of Fulani ancestry, and the Fulani are a nomadic pastoralist group that range across from Sudan to Western Africa, and a very small amount of Eastern African ancestry, which may represent remnants of the slave trade from Southeast Africa, across the Cape into the West. And then we see this Cape-colored population, which has nearly equal ancestry from the San, Southern Indians, East Asians, Europeans, and Bantu, very consistent with what we know about the history of that group. So I don't have time to get into this in detail, but I just wanted to mention that one of the other things we're interested in doing is studying how these populations have adapted to very diverse environments. So we have, for example, groups living at high altitude. We have groups with different diets, hunter-gatherers, agriculturalists, and pastoralists, and so on. And it's thought that if we can find those regions of the genome where they differ, that might help us define these genes or variants that play an important role in adaptation, some of which may also play a role in disease. And in fact, we did exactly this for a very common trait in East Africa, lactose tolerance, which is shown in light blue here. And we can see that many of the East African groups that have cattle also have a high prevalence of lactose tolerance. And we found several novel variants that regulate that trait, which are distinct from the variants that arose in Europe. And we were actually able to estimate the age of these variants to roughly 3,000 to 7,000 years ago, corresponding very well with the archaeological data for the origins of cattle domestication, which is a great example of gene culture coevolution. And then lastly, I'm just going to mention some very new data. I mentioned this recent expedition to Ethiopia, and these are some of the populations that we studied. And one of the traits we were interested in was skin pigmentation, because it's thought that that may be an adaptive trait. And so we measured the levels of melanin, and what you can see is the lower the number, the higher the melanin, or the lower the melanin levels, so the lighter the skin color. And here, the higher, the darker the skin color. And we found very significant differences, even in a fairly small geographic range. And one of the things now we're doing is looking at variation across the genome to try to identify the genetic variants that play a role in that trait. And then the last thing I'll show you is another really interesting variable trait, which is height. And we see the largest range in Africa, I think, for anywhere in the whole world, ranging from very short-statured pygmies to these very tall and thin Sudanese pastoralist groups. And this is another trait that we're quite interested in studying. And so the answer to a question I'm frequently asked by reporters, are humans still evolving? I would say absolutely, the answer is yes. And I just want to thank many people from my lab, many collaborators, funding institutions, and most importantly, the Africans who contributed to the study. Thank you.
Thank you, Dr. Tishkoff. It's a nice transition now to our next speaker, who I'll introduce, who's going to talk, if Dr. Tishkoff was talking about the variation among us, if you will, and how some of that plays into the diseases and the characteristics that we all have. Our next speaker, Dr. Frederic Bushman, is going to talk about some other sort of genetic information, if you will, which is carried around in us, but not in our own DNA necessarily, although sometimes it is, but some of those microbes that actually live among us and in us. Dr. Bushman's research centers on the microbes associated with the human body. Some members of this so-called microbiome cause disease, that's such as HIV, as Dr. Edelman was mentioning, or pathogenic bacteria, while others are important for health, such as gut bacteria, and those actually are helpful in terms of digestion. His lab looks at new technology, such as massively parallel DNA sequencing, to characterize and track such populations. He completed his doctorate at Harvard and has held positions at NIH, the Salk Institute, and he's currently here at UPenn School of Medicine. Dr. Bushman.
All right. So my title is Microbes in Our Body, Life Partners in Health and Disease. So what I'm going to be telling you is that there are really large numbers of microbes that live in your body. And it may strain credulity, but the way there can be such enormously large numbers associated with our bodies is that they're really, really small. And so let me start by just giving you a little background on the relative sizes of things. So this shows an aerial shot of the city of Chicago. What we're going to do in the next few slides is magnify in by factors of 10, getting smaller, 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 smaller. So each subsequent slide is going to be magnified by a factor of 10. So here's Chicago. So there we magnify in by one step of 10. We're starting to see the waterfront. Another step of 10, we now see the edge of the water and a grassy area and a road and a little bay. Now we're looking at the grassy area, and we can see something on the grass there. Each step now, each time I click the button, it's magnified by a factor of 10. So we've magnified by several factors of 10. Magnify again by 10, we can see it's some people lying in some grass. Magnify by a factor of 10, and there we can see some guy. So now we're going to magnify down into this person's body by steps of 10. So here's the person's hand. We magnified by a step of 10. Now another step of 10. We're going down into this guy's skin. There's a pore in his skin. We're now going down the pore. Again, each step being a step of 10. Magnify again, another step of 10. Another step of 10, and now we're at the level of cells. So we just did what? Something like 4 steps of 10, 10,000-fold magnification from looking at the guy's hand down to the level of the cells. So this is really small. Your body is comprised of something like, as Junhyong was saying, maybe 100 or 100 trillion of these cells make up your body. Now we're going to continue to magnify down. Another step of 10, we're looking at the outside of the cell nucleus. Another step of 10, now we're inside looking at the DNA all coiled up in a special way, as you can see here. And then another step of 10, we're down to the level of the double helix. And so here are the rungs in the steps of the helical ladder, which carry the information, the A's, T's, and C's, and G's that we heard about. So again, it was something like maybe, 3 or 4 steps of 10 to go from the size of a cell down to the size of DNA. So just to recap what I said, here's a scale that sort of shows the relative sizes of things. So the scale is in meters. Here's 1 centimeter, or 1/100th of a meter. Of course, that's about the length of maybe a joint in your little finger. So if you go down, magnify that by a step of 10, or 10-fold smaller, that's now a millimeter. Down a couple more steps of 10, you're in the sizes of animal cells, so 10 to the minus 5th meters. So if you keep getting smaller, you're in the realm of bacteria. These are the main components of your microbiome that I'm going to be telling you about in this lecture. So they're way small. They're at least 10-fold smaller than a cell. And then the viruses are even smaller. These are a form of microbe that can grow only when they grow on a cell. They're parasites on cells. And so 10, 100-fold down from the size of bacteria even. And then smaller than that, you get to the size of molecules and down to individual atoms. So your microbiome is the population of bugs that live associated with your body. It's comprised of bacteria, viruses, and other kinds of cells like archaea and eukaryotes. These are biologists' names for other forms of other parts of the tree of life. The human gut is home to something like north of 100 trillion bacterial cells and their associated viruses. This is substantially more than the number of total cells in the human body, which led one observer to comment, there are more of them in us than us. So the genome size, or the density of these bugs can reach something like 10 to the 12th per mil in the colon, per mil. And the genome size, the length of all the different types of DNA from these bugs all lined up, is at least 10-fold greater than the genome size of the human genome. And furthermore, many of these guys are unstudied. There are so many of them, they're hard to study. And furthermore, many of them haven't been grown outside the human body and subjected to careful study. So many of them are very little studied. And they're distinctive microbiomes at each body site. The bugs in the gut are mostly different from those in the lung and different from those in the skin, the mucosal surfaces, and so forth. And so this shows pictures of some of them. In cheerful false color, the bacteria of your gut. Some of these are nasty pathogens like Clostridium, Salmonella, others more benign. And so returning to the question I asked at the beginning, how can there be so many microbes associated with your body? Well, the answer is they're really, really small. Okay. For the most part, there are occasional creatures that are associated with your body that aren't necessarily that small. Many of you will know about the nasty intestinal worms that can live in your gut. Unfortunately, some of those guys aren't small at all. Here's one you might not know about. This guy is called Demodex. It's a cute little worm, an arthropod that looks like this. It actually lives in hair follicles of animals that have hair. Here's some Demodex's little behind sticking out of a hair follicle, and here's the hair coming out. Something like 33% of children and 66% of adults have Demodex living in their hair follicles. Bet you didn't see that one coming. Okay. So host microbial mutualism. You can think of your microbiome as an organ that's contributing to your health, or at least a lot of it is. The bacteria benefit the host by assisting in digestion. Many of the substances in food that you eat couldn't be digested if not for the contribution of enzymes encoded in your gut bacteria. They stimulate immune development and maintain immune tone. So you can make animals that don't have any associated microbiome, like mice, and their immune systems don't grow normally, their guts don't grow normally. Turns out that when you're not infected by some nasty bug, like a virus or pathogenic bacteria, your immune system is not sitting around inactive. It's sensing the antigens of your normal microbiota. It's responding to them. It's changing the composition of the community. And that's to the benefit of both of them. Maintaining healthy immune tone is essential to your health. If it goes wrong in either direction, it can harm you. If your immune system is insufficiently active, then bugs that are normally benign can become pathogenic. And that's what we see in diseases like HIV, immunodeficiency. Going the other way, if your immune system is overactive, that can cause autoimmune disease, like inflammatory bowel disease. So this whole process of immune tone intimately involves your microbiome, and it's essential for your health. Your microbiome can also contribute to metabolizing drugs. Again, some of the enzymes that degrade drugs and make them go away as they pass through your body are contributed by your microbiome. And the development of correct blood vessels in your intestines is actually dependent in part on having a correct microbiome present. Going the other way, you benefit your bacteria, you provide a safe environment for them to live in, and your body even provides nutrition for them that they can eat in lean times. So the study of the human microbiome has been revolutionized by the new sequencing methods that Junhyong mentioned. So the way to study these microbiomes now, overwhelming first choice, is to take samples from a part of the body you want to study, from the lungs, from the gut, from the skin, purify DNA, and then apply these marvelous new deep sequencing methods to determine sequence. You can then use computers to align the sequence fragments that you get to databases and use that to count the kinds of creatures that are there, determine their nature and their relative abundance. So the types and proportions of microbes at different body sites can be determined by DNA sequencing. And here's a little snippet of the DNA helix. So this area is turned into like a commercial explosion. There are now a whole bunch of companies that are marketing instruments that have just tremendous sequencing capacity. Something like, this is probably already out of date, but the Solexa and ABI instruments can generate something like 100 million sequence reads of 100 bases in a run that takes a few days, maybe a week. So we're north of we're getting tens of billions of bases of DNA sequence in a quite short time. Quite recently, we were rushing. We wanted to get a bunch of data done for a grant application, actually. We sequenced north of 1 billion bases in a week. This is just absolutely incredible to me that this is possible. It's like there's space aliens secretly behind all this technology or something. And so this is a gigantic race now among a whole bunch of companies with extremely clever chemistries and methods for DNA sequencing. And so this explosion that's happening now is showing no signs of slowing down. So for those of us who've been interested in these kinds of issues for a long time, it's just remarkable to have this new set of tools that we can apply to these sorts of problems. It's really like being a kid in a candy store. And so there's my daughter, Jocelyn, who's here, actually. She's cheerful because she's going to get to eat the candy after putting up with my photograph. Okay, so let me tell you a little bit about results that we've had from sequencing. So we did one experiment to try to understand the role of diet in controlling the human microbiome. Since this comes up in many disease states, as I'm sure you know, bad diet is linked to all sorts of disease states. So we wanted to try to understand how it was influencing the human microbiome. We hired 10 healthy volunteers, randomized them to a high-fat or low-fat diet, then locked them up in the hospital at Penn for 10 days and fed them either a high-fat diet or a low-fat diet. We sampled poop from these patients. We sampled all kinds of other microbiome sites on their bodies, including tissue biopsies. And then we applied deep sequencing. We purified DNA from all these specimens, deep sequenced to see what kind of bugs were there. Again, going north of 1 billion bases over this whole experiment, just incredible amounts of DNA sequence. This gives you some idea of the kinds of data we like to look at. So this shows one of the subjects. These are 10 time points where each bar graph shows one time point. So we've gotten DNA sequence information, aligned it to databases, read out the kinds of bacteria that were there, and then assembled it into this kind of bar graph. And over here is a key showing all the names of lots and lots of different kinds of bacteria specified by the DNA sequence data summarized here, and then the different time points are shown here. And so you can sort of see by eye that the gut bacteria we see when this person first comes into the study is actually rather different than the gut bacteria that they have later on the defined diet. And so by doing a whole bunch of this kind of data over all the subjects, we can get to significant data saying that the human gut microbiome changes over when you have a dietary intervention in as little as 24 hours. We can measure such changes. And so there's lots of applications for this kind of work. We're trying to optimize diet therapy for inflammatory bowel disease. Turns out that, particularly for children with inflammatory bowel disease, one of the treatments for the disease is to put them on a simplified diet. And so we're studying now the linkage with the microbiome and its effects on, and how the elemental diet affects that, with the idea of optimizing therapy. We're also seeking to understand dysbiosis or adverse microbial communities in various disease states like HIV infection or asthma. And we're seeking to understand dysbiosis or unhealthy bacterial communities in response to other kinds of environmental factors such as smoking. And I can tell you that smoking is changing your lung microbiome in a way that's probably bad from other data. But that's one piece of the community. That's the bacteria. Let me now briefly end by reviewing another topic, the fascinating but disturbing topic of the viruses that are in you right now. So many of us are familiar with the persistent viruses that infect people, like the herpes viruses, for example. A substantial fraction of the adult population is infected with these different herpes viruses, some approaching 100%. There are other kinds of viruses that are benign and really wouldn't be known about at all if not for the new sequencing methods that are very common in the human population. Others, persistent infections, very serious, HIV and HCV afflict something in the range of 1% of the human population, of the US population. But that's just the tip of the iceberg. There are viruses that come and go that are very common. 90% of the adult population or elderly population has been exposed to parvovirus. There are these other kinds of benign viruses that probably come and go. If you have a cold, there's a good chance you have one of these viruses. And then sometimes we deliberately infect ourselves with viruses like the Sabin poliovirus vaccine or the smallpox vaccine, vaccinia virus. But still, that's just really the tip of the iceberg. So there are viruses in the human gut that prey on the bacteria that live there that are present in absolutely remarkable numbers. So Sam Minot in the lab has been studying these kinds of populations. He finds something like 10 to the 10th to 10 to the 11th virus-like particles per gram of human poop. So here's the, one of those viral preps stained for DNA, and you can see it's like stars in the sky. He's diluted it way out, and still there are many, many, many viral particles present per field. This is true of all the samples we've studied, so I think it's fair to say that your poop has something like 10 to the 10th to 10 to the 11th virus particles per gram. And here's what some of them look like, the bacterial viruses. We actually studied these over the controlled feeding experiment I mentioned for bacteria, and we can show that a dietary intervention is having a strong effect on the viral populations as well as the bacterial populations. So we have a very clear picture of how many of these viruses grow. This is a picture of phage T4, one of these bacterial viruses, infecting a bacterial cell. This movie is wonderful because this isn't some cheesy artist's reconstruction. This is a real summary of structural biology data coming from the laboratory of Michael Rossman and others. There's a real, the structures of each of these proteins have been carefully determined, and the picture built up over lots and lots of structural studies. And so you can see the pinning and attachment and pinning by this phage. It's now injecting its tail tube, injection tube, breaking through the cell wall, the periplasmic space, and it's injecting its DNA. So that virus DNA will multiply, generate many copies, explode the cell, and leave several hundred copies in the next generation. And so my last slide, here's saying that even what I just showed you is still the tip of the iceberg, in that there are viral remnants in the human DNA itself. The DNA that's present in every one of your 10 trillion cells has a whole bunch of virus sequences in it. So the human endogenous retroviruses, as they're called, or HERVs, are viruses that donated their sequences to the primate lineage leading to humans. They comprise something like 8% of the human genome is comprised of these sequences. If you want to look at this yourself or anything else about the human genome, you can go to the UCSC Genome browser, type that into your computer, and it'll bring you to this site. Here's one of the human chromosomes, chromosome 11. We're at this little place on the chromosome where this little red box is, and that's shown here. Here's some genes that happen to lie in this region. And this track here where it says LTR is actually viral remnants that you can detect over this part of the human genome. So 8% of your DNA is fragments of viruses. Many of these are inert, they're not doing anything, but some of them have been recruited to make new genes that are unique to humans. And so these viruses are really a part of you. They're contributing to your genes. So the main points for my lecture, you can remember, yes, this is a bacteriophage made out of Lego by my son Marshall and I working together. But the main points of this lecture, humans are hosts to vast colonies of microbes. Some help, some hurt, some just hang out. And lastly, pieces of microbes are pieces of us, right down to our genes themselves. And so I'll stop there and continue with the program.
Thanks very much. I'll treat this gingerly, I think. So I'll just stand here. Thanks very much, Dr. Bushman. I suppose you could always have a good news, bad news thing about what's infecting you. I'll choose to think about the fact that given my particular hair follicle situation, maybe I have less of the Desomox, am I remembering? Demodex, excuse me. Unfortunately, I probably have more of the other things living the life that I have. I wanted to, we're going to bring up now two other scientists to join us in a discussion, and I wanted to introduce them and to have all of our speakers as well as those joining us in the discussion join us now at the table here.